All right, so, well, thank you all for being here. This is welcome to the Center for Reproducible and Biomedical Modeling Seminar Series. This is the summer of 2024 edition. Purpose of the series is to discuss various challenges that arise in biomedical modeling. Presenters will put forth methods, tools, principles to address those challenges, and then at the end we will discuss them. I'm your moderator today, Michael Koshin. A few things to get out of the way here. Schedule. So we got 2 presentations in this series for the summer. First one's today, Dr. Pedro Mendes. He'll discuss fair data principles as applied to biomedical models. On July 12th, we have Paul Jost. He will discuss PyPesto, a platform for parameter calibration. Format of these is both of them as a 50-minute presentation followed by a 10-minute group discussion. Please mute your mics if you're not talking, and if you have any questions, you can put them in the chat for later. We'll be monitoring that. Today's speaker, Dr. Pedro Mendes. Since 2020, he has been the director of the Center for Cell Analysis and Modeling at UConn Health. He's had a long, productive career developing, promoting models, biochemical systems, and the tools to study them, including authoring Gepasi, the predecessor to the very popular biochemical network simulator COPASI. Today, Dr. Mendes is going to talk about fair data principles as applied to biochemical models. And with that, the floor is yours.
Thank you, Michael. Let's see if I can share my screen. Okay, hopefully everybody can see it. Okay, so I'm going to talk about an exercise that it came around rather organically. This has all been published, so you see there the reference for the publication. So all I'm going to talk about has already been published, and all the materials are all public, as you will see. That's actually part of the whole case that I'm going to make here. And essentially, I'm going to be talking about or showing with a very detailed example of issues about reproducibility and fair principles when we use computational modeling. Okay. So the motivation for this came from a number of sources. The first one was MD PhD student we had here. Actually, I was in her committee, and Lauren Marazzi, and she wanted to use this von Dassault model, which you will hear in detail for the rest of the talk. This segment polarity network model to use as an example for the application that she was creating. However, the problem was the model was not available in any model database, and so we did a little bit of an exercise, and as you will see, she did find out one version in SPML, which she then used and adapted and used. But in the meantime, I got a little troubled because this paper is very famous, as I will detail in a moment. And so I've also been collecting classic models in COPASI format for teaching and examples and other things. And I thought this would be a cool one to have. And also for a project that I have been working on and off with Eran Agmon, who's here in the audience. I thought this model would be an ideal one for testing, basically an approach for composition of models with larger models with smaller OD models in a kind of a multi-scale approach with his Vivarium software. So I really wanted to get this model running in COPASI. Now, I'm going to be talking about fair principles, and just to make sure everybody's on the same page, here they are, as they were proposed in the 2016 paper, now very famous paper. So their principles were for scientific data management, and for many of us, probably models can be seen as really mathematical objects, maybe programs or functions. Not really, we don't think of them as data, but they also can be seen as data. They're actually complicated structured data objects. And as such, I think they also should abide by the FAIR principles. So they should be findable, that is, they should have data and metadata assigned a globally unique and persistent identifier in some repository somewhere. And as you have seen already, that this model I'm going to be talking about failed in that respect. They should be accessible, that is, they should be accessible by their ID in some free open protocol. Again, we couldn't get that. They should be interoperable, so they should be expressed in a formal language that is broadly applicable for knowledge representation. In this case, as you will see, that this will be SBML and SEDML and etc. And then they should be reusable, so they could be associated with a detailed provenance and meet community standards, etc. So those are the four principles. There is one other, which is in the title of the center for which I'm giving a talk now, which is reproducibility. And as you saw, FAIR, the R is not for reproducibility, it's for reusability. But reproducibility is very tightly connected with the FAIR principles. And as you most likely have already heard or read in various publications, there is what is perceived as a reproducibility crisis in science. And surprisingly, this is also affecting computational biology or computational science. And now in a moment, I will say, well, you could think why I say it's surprising. Because in principle, if everything is written down in a piece of code that is executable, then it should be very easy to reproduce. You just run things again. Now, before I even go any further, I will also try to just a little bit with a series of concepts. The word reproducibility has been used in many different ways. And in fact, other words have been also used in sometimes overlapping meanings. For example, replicability has been used as well as reproducibility. And in fact, there's even a nice paper that talked about the fact that this term is very badly described. I tend to adopt the description from Goodman et al., which they coined 3 different types of reproducibilities. The first one is reproducibility of methods, which means that someone else should be able to exactly reproduce an experiment using the same methodology and the same data. So that's really, really doing the same thing again in someone else's hands. The second one is reproducibility of results. That is, someone should be able to obtain similar results in an independent study, applying similar procedures, but independently. And this is, as you will see, what I will be attempting to do here. And then finally, the strongest one for the point of view of epistemology, reproducibility of inferences. And that is, you are able to reach the same conclusions in an independent replication, potentially following a different methodology. And that we will not do here, but that's basically arriving to the same conclusions by a different set of experiments, essentially. So you reproduce the inferences, not really the results. So in this case, we're going to focus more on the first two, particularly on the second one, reproducibility of results. Because as you will see, I don't have the original software that was used to create the results, and therefore I'm going to use similar procedures, but not exactly the same. So to describe the model a little bit, this is a very classic model. It appeared in 2000. There's a lot of papers published. At that time, 2000 has been seen as a year, as a marker for the beginning of systems, of the modern systems biology. For different reasons, I won't go into this. If you read my paper, I elaborate a little bit about it. But this paper is probably one of those that was very widely discussed around that time, and it has since been cited many times, more than 2,000 times, I believe. This paper covers a model, a computational model of the Segment Polarity Network. That is a network of genes, which is represented here. Network of genes that are expressed and that are required for Drosophila embryos to form these patterns that you can see here, these green lines. These are patterns that you can observe by staining for certain markers, certain proteins. that are expressed in some parts of the embryo, in some cells of the embryo, but not in others. By the way, this is an advanced stage of the embryo. At this stage already, cells have their own plasma membranes. So they're actual cells. It's not the syncytium anymore. That was in earlier stages. So there's actually a lot happens in the development of Drosophila egg, and this is already at the later stage. So these cells are formed, and they essentially express different levels of these genes, and they form a pattern. And these patterns will then be what marks the segments of these insects. So in this case, the fly. And each segment will then lead up to a pair of legs or wings or whatever. And segmentation is a common process in insects. So the model includes many cells. Each cell has this network of reactions. So by the way, this line here is a membrane from one cell, and that's the neighboring cell. That's cell next to it. There's a number of genes and proteins. The lowercase symbols are the mRNAs, and the uppercase symbols are proteins. They have a series, of course, the RNAs express the proteins. The proteins sometimes do something else. For example, the CID is a precursor for CN. There's a cleavage here caused by PTC, which actually is a membrane protein. These proteins here are membrane proteins, hedgehog, patched, wingless, engrailed, cubitus interruptus, this CID that ends up being CN. So these are the genes that are involved here. And there are also interactions between the neighboring cells. So this wingless protein that is on one membrane can actually pass to the other membrane of the other protein, and it affects expression of its gene. Patched is a receptor essentially, and it receives hedgehog. So hedgehog binds to patched, and this is the hedgehog patch complex. It's represented here on one side, but of course it's the two membranes have to be really close by. And they can be, both cells have both patched and hedgehog. So the hedgehog on one side binds to the patched on the other, and vice versa. And then in this model, they represent cells as hexagons, which as you will see is a really bad idea. I will tell you, never try hexagons. They're a piece of the work of the devil. So these hexagons, basically, each cell has 6 neighbors that are mapped. So there's not just one membrane, there are 6 membranes. And these pools of proteins that are on the membrane, they actually have 6 of them. So each side of the hexagon contains a full independent pool of these proteins, and they can diffuse from one side to the other. So there's also lateral diffusion around the membrane, and there's diffusion in the case of wingless from one membrane to the one neighboring, and then these binding interactions between patched and hedgehog. So that's kind of the model. And this paper is famous for the fact that it basically expressed the concept that the network gives a lot of robustness to the physiology of this embryo. And in the sense that they sampled a lot of values for the kinetic parameters, so there's a bunch of kinetic parameters and they don't know the values for those parameters, and therefore they just sampled them, did really very large scale random sampling of values and tested the models, and they found out that in a surprisingly number of cases, surprising to them, there was always a pattern forming, that they could reproduce the same pattern of genes really with many different sets of parameters randomly sampled. And that's why this paper is famous. That's the message. Now, I wanted to reproduce the model. So the first thing is I looked for the software that they used to create this model. And that's a software called Ingenue, which was, they described it in that paper. They described it in a paper 2 years later in detail. This paper here is a good description of the software. And as you can see, it's here, screenshots of the software running on an Apple Macintosh, likely. A few years later, in 2009, a different author published about the same software in a book entitled Methods in the Methods in Molecular Biology series. That this was edited by Ivan Mali. And however, that software doesn't exist anymore, or it doesn't exist in an accessible way. So I followed the paper. The paper said the software could be downloaded from ingenu.org. So no, ingenu.org doesn't exist anymore. But then the 2009 paper said that it actually could download it from ingenu.com. So there was probably some change of minds of whether they wanted the software to be open or commercial. I tried ingenu.com. That also doesn't exist anymore. So I thought, okay, let me go to the Wayback Machine and try and get this stuff some other way. So I did find the pages on the Wayback Machine. And unfortunately, in the Wayback Machine, there's a link to download the software. But if you click it, the Wayback Machine says, oh, we never cached that page. So there's no software to download, even though I could obtain all the web pages. They were all archived in the Wayback Machine. So I couldn't get the software at all. Now, having said this, since then, in fact, by today's host, sent me a copy that he had downloaded earlier on. Now, I figured out later on that this is probably tied up with authors retiring or etc. But I found out that unfortunately it was not even retirement, was sadly the senior author of the paper, Gary Odell, had passed away in 2018. And that's, I think, when his lab's web pages lapsed and nothing else was to be obtained. Now, I could have tried to contact the other authors. I decided not to do that on purpose, exactly to try and reproduce the model and see how much can I reproduce it just from reading the paper. And that's what I'm going to do now. Just in the meantime, this paper, as I said, has been very widely cited, and it has also been reused and reproduced. So part of the fairness has been already demonstrated before. So these are all of the, I followed all the 2000 references, and these are the ones that actually did something with the model. So these actually reused the model or coded slightly different versions of it. I would point out that it's not just ingenu that doesn't exist. Some of these papers were published with, or some of them we don't know how they were run, because the papers don't say how the software was, how the calculations were obtained. Some were written with bespoke C programs that were never distributed. They don't exist. We don't know what these C programs are. Some of them were made with some other software like LittleB that also is no longer available on the internet. You cannot get LittleB anymore. A few others were done with Mathematica and MATLAB and Python, and then some other software like Sloppy Cell that you can still get. Well, I tried to make my COPASI version. Lauren Marazzi had adapted one SVML file she discovered on the web, which actually comes from I believe this Daniel's paper. This paper here from the Setna group. They actually have that online, and it's a SVML model. Unfortunately, it doesn't encode the network. It only encodes the ODE, so it has no stoichiometry, only just the ODEs. And so that's, to me, as an example for coding, was really a bad one. So I decided to still go ahead and move forward. Some of these models implement different versions. So some are Boolean networks. Some others use a square grid of cells rather than the hexagonal. And I could tell you it's a lot easier to do square grids than hexagonal grids. I understand them very much, having done the hexagonal grid. So here I go. There's the model is described in supplementary data. Surprisingly, the supplementary data is still available from the Nature website, even though it's 24 years after the paper has been published, which is good. Not all journals have that. A lot of journals, old papers, the supplementary data has disappeared and is no longer available. So the paper had a bunch of equations. It had textual descriptions, tables with parameter values, fuzzy values for the parameters. As you can see, what they do is give ranges, and they say these things are sampled within the range, but they don't give all the details. But anyway, going through this, I was able to reconstruct the model. This actually I already mentioned. The model is based on hexagonal arrays of cells. And they say in their software, they can make as many rows as they want, as many columns. By the way, the columns have to be multiples of 4. They have to be at least 4 cells side by side. Otherwise, there's no patterns. And they were organized as a toroid. That is, basically the edges are connected. So this edge here is connected to that edge over there. If you just think mentally how this thing folds on itself and forms a donut, that's basically the connections. And so I ended up trying to reproduce the same thing. I ended up using a 1x4, so a row of 4 cells, which is also what they say they used in the paper. That's the minimal number of cells that forms a pattern. It always has to be an even number because hexagonal arrays don't deal well with odd numbers of cells because, as you can see, they're have to pair in twos. But so I ended up doing 1x4 as for the example. This is my version in SVGN diagram of the network. It's a little bit more formal. So I'm trying now to use community adopted standards, and that's really to try and make the model fair. So if you remember, I believe in the accessible, let me go back to that. One of the letters of FAIR is about interoperable, right? So by using formal languages, I'm making the model interoperable. So if you look at their diagram, these diagrams, you don't know what these circles mean and the arrows, and you know why some arrows have a circle and others have a pointed arrow. There's no convention that defines that very well, but in this version that I now present to you, it's using the SVGN standard where these symbols have well-defined meanings. So this is a symbol for an mRNA, and that's a symbol for a protein. The colors are actually arbitrary. I could have kept them black and white just without colors, but I decided to make it more obvious that the RNAs have a yellow background and the proteins have a green background. I've added the hexagon showing the next neighboring cell. What I did not show is that this protein exists in all 6 sites as different pools, and that it can diffuse from this side to that and from this side to that, and vice versa for all of them. So this is not a complete diagram of everything because it would not be legible if I put all the reactions. Okay, so what I did is that I had to create a Python program to build this, but I think I'm getting a little bit ahead of myself. I'm going to first describe their results and then get to that. So because when they constructed this model, because they did not know values of the parameters, they said basically, well, we don't know parameter values. We're just going to do a very large random sampling of all 48 parameters of the model and initial conditions, and then find out which ones, what proportion of them end up with the pattern that we want, that is the correct pattern observed in Drosophila eggs. And they did those samplings with different sets of initial conditions, as you could see here. For example, CRISP is a set of initial conditions where they put the values of certain of the genes and proteins in the 4 cells in different patterns. It's a pattern that already forms a CRISP edge. Degraded is similar to the patterns that we want, but with lower values, and they don't express these very well, etc. I'm not going to go through all of them. They are described, but they're not described with a lot of detail. These letters, M means medium value in the range, H means high value in the range. They don't tell you between what and what is high and mean and low. So I had to do a bit of guessing around here. And then this is how long they do the simulations for. Different lengths for different of these initial conditions. And then this is the number of samples that they tried here and how many actually led to the pattern that is the correct pattern that is observed in Drosophila. And then they tell you proportion, how many, the hit rate. So how many patterns out of the sample were correct. So, and their argument is that 1 in 201 is a very high number, given that you're sampling in 48 dimensions. I'm not going to, my talk is not about this. I tend to disagree with it, but I'm not going to make a case about that. That's not the point of this talk. And really, I don't want to get in there. But I think that, just my opinion, I think that I don't know why this is a small number. There's no good argument about the argument that they put forward is not a good one, in my opinion. They then display their results this way. So they plot all the samples that generated the patterns, and they plot it in this radial representations, these radiographic representations, where each of these spokes here is one of the parameters. And this part here is high value, that part there is the lowest value, and there's everything in the middle. So they normalize the parameters such that, so one line is one model, and that line will connect to each of the spokes to whatever value that parameter has. So then they plot all of the ones that produce the pattern here to try and see if there's any observable patterns on the parameter values. And you can see some things. For example, this parameter here is never very high. Probably high values of this parameter do not generate the pattern. But other than that, you can see that they are all over the place. Now, this one is for a different initialization, so that in that case there's less hits, so you see a little bit better. But these are their results. This is what they produce in their paper. And this is the claim of robustness. As I said, I'm not really going to focus on this. It's written in their paper, and I discuss it in my paper too. But what I care about is what I'm going to do next. How did I reproduce the model? So if they had used a square array of cells rather than hexagonal, I could have done it directly with COPASI. COPASI has a special tool to replicate a model into an array of compartments. So you could create an array of all the compartments and mention which reactions exist between the compartments or create those. And then I could have done that straight with COPASI, but not with an hexagonal array. So I thought this would be a good idea to write a piece of Python code. And fortunately, Frank Bergman created Basico, which is a high-level Python API for COPASI. So when you use Basico, basically you write Python, you import Basico into it. And that essentially means that you're going to be using COPASI. Basico is just a stub to the COPASI binaries. So when you use Basico, you're actually using COPASI in the backend. But you do it from your Python code, and so you never know that you're using the C++ program, the binary. So I used Basico. This was perfect. And I had to do that because of the hexagonal array. Now, this is a piece of the code I don't want you to read. I just want to highlight one thing. This is the coordinates of the cells. And if you can see, there is no way to address hexagonal arrays without having to have if statements, because some cells like 0, 0, and 0, 2 have neighbors up and down, where other cells like 0, 1, and 1, 1 have neighbors down and up. And so what are the sides neighboring to this cell are not the same numbers as, so for side 6, 0, 0 is neighbor to side 3 of 1, 1. But for side 6 of 0, 1, it's neighbor with 0, 2. Now, 0, 0 to 1, 1 is not the same pattern as 0, 1 to 0, 2. So you cannot index this. You have to put an if statement. Now, this one is the same as that one. And, so the odds are equal to the evens, and the evens are equal to the odds. And that happens with 2 of the sides. 4 of the sides, you have no problem, but 2 sides, you always have to do this. And there's I discovered that there's a lot of literature written about how to index hexagonal arrays, and it's really a work of the devil. Probably because 6 is not a power of a prime number. And I shall just stop there. Anyway, I wrote that there's basically, it creates the grid, does everything, creates all of the species on all of the cells and the reactions that are needed, et cetera, and then produces a COPASI file that I can load into COPASI and produce results. That's essentially what I did. Now, when I was encoding, I found out there was some, even though I will show you, I could reproduce the results, there were some problems remaining. They did not feature this parameter in their model, in their description, and in the supplementary data. That parameter is allegedly equal to another one. It's the lifetime of one protein. It's a EWG. It's a protein that gets cleaved into another one. And well, no, this one is in the extracellular membrane, and that one is in the cytoplasm. And in their description in the supplementary data, they have the same half-life. But clearly, given the plots, they don't, because they exist as 2 separate parameters. So there was no description of one there, but reading the other papers that were written about it, particularly other papers from their group that were written about it, I did find out that they basically were 2 parameters rather than 1. So one is solved. The next thing is that they say they sampled all 48 parameters of the model. However, the model has 53 parameters. So which 48 out of the 53 were the ones that they sampled? And again, looking at their figures, I could identify 46 of them were obvious, but then there could be a number for the other remaining, the other remaining 7 could contain the missing 2. And it was only a picture from a plot in the 2002 paper, a later paper they published, that I identified what are the 48 parameters. So this is a problem. Here are the problems for reproducibility. You don't describe everything in your model, no matter how much careful you are. And I have to say this paper is really good because I did manage to replicate it. And the supplementary data has a lot of detail. Still, it's very easy to have something missing, which stops anyone else from replicating the model, or at least makes it very hard. The ranges for the parameter samplings, they are provided in a supplementary table, but they only include ranges for a number of them. They miss ranges for these 2 parameters, so I have to come up with them some way. I did find some network files, not the software in GNU, but the files that are fed into it in GNU. Again, I found them on the Wayback Machine. And from those, I thought that the parameter range should be 10 to the 3 to 10 to the 6, but it could also be 1 to 1,000, which is mentioned in the Kim paper. So there was a question whether it's this one or that one. I tried both, and it seems like the 1 to 1,000 is the one that it's the values that seem to be correct. However, as you can see, this was mentioned in the paper 9 years later than the original. Then they use a score function to identify parameter sets. I could not replicate that because, again, they don't specify everything in that function. I had to use a function which is less restrictive than theirs, which means that in my results, I may have identified more hits than I should have. So my data is potentially a little biased because I don't have the same scoring function because I couldn't reproduce it. And then finally, the initial conditions that they prescribed, again, they mention the ranges like 20 to 60% on the range, and they don't tell you what values they use in that. Is it randomly sampled? Is it a number in there? So I ended up using this for the ranges they mention. So when they say it's less than 20%, I say 0.15. When they say this for the initial conditions, when they say 20 to 60, I use 0.4. When they say 60 to 100%, I use 0.9. By the way, all the parameters are scaled. This model is completely scaled. Everything is 0 to 1, except the protein that has 6 sides, which of course the sum of the 6s end up being 6. So everything else is very well scaled within. And you will see later on that was actually a problem for some simulators. This model has only one time scale. Everything is on the same time scale. No stiffness. So these were not correct conditions. I don't know if I'm replicating exactly what they did. Oh, and then there's another problem, which is not really something that they even discuss, but it's something that you immediately will come across if you do random sampling of any model that has Hill equations. And this is because, and you may not know this, but anything that has an exponent in computers, if the base is not an integer, the numbers are calculated as, so if you have a base raised to an exponent, this is calculated as e raised to exponent times the log of the base. This is how computers calculate this. You may not know it, but that's how your computer calculates that. And the problem with this is that if your base is negative, this doesn't work because you cannot have log of a negative number. And in principle, the base here are concentrations, so they should never be negative. But if they are very small, if they're a number close to 0, because of the nature of most differential equation integrators, they're predictor corrector methods. They make predictions and correct them. Sometimes the prediction undershoots 0 and goes to negative. And then it'll be fine. It'll be corrected to a correct value. Except that when that is done and that base is, even if it's a very, very small minus 1 times 10 to the minus 10, it's a very small negative number, but immediately generates another number, and everything else in the computer becomes another number, and your that simulation ends up with no results. So when you do random sampling, this happens at quite a frequency. I found out that this happened with a frequency of around 30%. 30% of the random samples would, at some point, one of these functions, there are many of in the model, would generate a none and would produce nothing. So I decided to use a so-called protected version of this, which is I calculate base raised to exponent as being the maximum value of that, or if that is not calculatable, an epsilon. And an epsilon is a number that I use that is bigger than 0. So if the base is a little bit smaller than if it's smaller than epsilon, I'm going to use epsilon as the exponent, as the base. So if my epsilon is 10 to the minus 10 and the base was to be 10 to the minus 20, I'm going to use 10 to the minus 10. So that makes it a small error because this number would be small, but that avoids the not a number calculations, then logarithms of a negative number, and therefore Hill equations like this become Hill equations like that. And my software uses this, and I used a value of 10 to the minus 80 as the epsilon, which is really a very small number. You could say this is 0. And that reduced the errors from 30% when I had just that to about 0.1% of the samples. By the way, still 0.1% still work because even 10 to the minus 80 is not that small. Some cases it will still generate. So, but I can live with that. 0.1% of the samples were broken and I just did nothing about it because the other 99.9% were fine. And these are my results compared with theirs. So I did the samplings for all of those initializations with the caveats that I'm not sure they're exactly the same. These are their numbers, their hits, and the number of tries and their hit rate. You could see here the problem of the logarithms, because I tried 240,000 samples, but only 239,272 were produce results. The others didn't produce any results because they had that negative number, that problem with integration. But if you look at the number of hits, they're not that far off. Remember, I don't have the exact details of everything, and these are random samplings of a model with 48 dimensions, and we're only taking 240,000 samples, which is not that much in 48 dimensions. And so even the hit rate is not that far off. This is probably, the second one is probably the worst difference. So their hit rate was 1 in 5,000. Mine was 1 in 35,000. But the caveat here is these are very rare events. We are trying to sample very small numbers. And so I would expect that if these values here, other than 750,000 simulations was, I don't know, 750 million simulations, I probably would get a better approximation. The approximations are better when the values are higher, right? So this last one is 1 in 46. In my case, is 1 in 39. It's not that different. So I think these are pretty decent numbers. I basically come to the same conclusions. If I had to say qualitatively analyze my table to their table, I would not probably come to conclusions different from theirs, except I don't think this is surprising personally, but anyway. And I even was able to reproduce their figure 2A, that one that I showed you before. This is with what they published in the Nature paper. This is what I created with my results. So you see the same parameters. By the way, this took a long time to create this figure with GNU-plot, but I managed to basically have it almost exactly the same. So even the parameter I mentioned before, this KCEN, same thing that they found, that in most cases when this value is high, there's no pattern. So if you only select the samples that give you the pattern, you never get samples, or you have very few samples that have high values for KCN. And there's probably a reason for that, but it's outside of the scope of what I'm going to talk about. But right, so I managed to reproduce. I would say I did I was able to do a reproduction of the second level, reproducibility of methods, I believe it was. Let me go back to reproducibility of results, exactly. So I'm sorry, I have to go through the slides all over again. And I'm aware that I should be wrapping up. It's getting closer. Okay, so now for the findability and accessibility. So I now have model files that are COPASI files. Of course, I can export them as SBML, ZML, put them all together into a NOMEX file. I did that. My code is in GitHub. It's open. Anyone can go and check my Python code that produces the model. So the model is created by a Python file. Then that model you can load in COPASI and do the samplings. So that contains the scripts, the model files produced by the scripts, figures, processed results are all there on GitHub. Not all results, because GitHub, some results were too large. GitHub didn't like me to upload them, but everything fits in Zenodo. So I also added a Zenodo entry. This DOI has all my results. They're there for posterity. Everyone can go and download that. That includes also the code, the scripts, everything. Raw results, the whole kitchen sink is in there. And then I also exported my model to biomodels. I uploaded it to them, and they were very happy because what I did was exactly what they would have done, which is replicate the model. That's what their curators normally do with the paper. So they actually took it and added it as in the curated models version, and I got labeled as the criminal who curated this model. I did reproduce, so all the model files are there, and also my figure 2, the data and the code to reproduce figure 2 is there. So this would allow findability and accessibility. So now if you look for this, for a model, for the von Dassau model, you find my uploaded SBML file, or perhaps the Zenodo repository with everything, and you will be able to use this not just in COPASI but in other software. Okay. So that's the Zenodo entry. That's got a DOI, etc. This is the this is my GitHub page. This is the biomodels entry. And now for interoperability. So now we're up to the I of FAIR. And I want to demonstrate the model can be run on many different simulators. So I thought, okay, I'm going to set up a time course of one sample. I just pick one that ends up with a pattern because that looks nicer. And I export that in OMEX format and also SBML. And I thought, well, and then I run it in COPASI. And this is the time course. This is basically the 4 cells here. You see 1, 2, 3, 4. That's 4 hexagonal cells side by side. These are the patterns of the various mRNAs. They form a pattern, as you can see. In cell 3 has a different value than cell 2. By the way, this is where the segment would be separating, is in this where this dashed line happens. So 2 cells are on one side of the segment, the other 2 are on the other side of the segment. This is the protein levels, the uppercase variables here. So this is the time course. So now running this on other simulators. Well, I thought this was a very good use case for run biosimulations, another product of this center. So I tried to do that. Since I had the OMEX, I just had to upload it into run biosimulations and get it working. And this was the outcome. So Amici worked. Amici is a package that actually is MATLAB package, but run biosimulations created a wrapper for it. I think they have a container that has everything. That one managed to run, produced a time course that matched what I did in COPASI. So there's interoperability already proven, because 2 pieces of software could use the same SBML file and produce results. With VCell, I got wrong results. I got results, but they were wrong. And with LIPS SBML sim, I also got wrong results. I'll get to that in a moment. And then all these other 4, Tellurium, Pisces, Gillespie, Pi2, and MassPi, they all failed for different reasons. Tellurium failed with a CVOD error. Pisces couldn't do species assignments, and there are species assignments here. Gillespie 2 could not read the ZML, and MassPi cannot read SBML level 2 version 4. Okay, so VCell. My colleagues here produce VCell. I think I saw at least Jim Schaff is in the audience, I think. So I just had to go next door and bother Jan Moraro. Jan, why does this not run in VCell? And he found out there was a bug that had creeped into VCell, which he managed to fix. So I got a file that worked. So I used that one, and then I got the same result as Tellurium. It gave me a CVOD error, character convergence failure. But Virtual Cell has many different integrators, so I picked a very old integrator that's there, the Adams-Moulton method, which was coded, for those of you who may remember this name, was coded by John Wagner back in the 90s, I think. And that actually produced a result that is a success. The results match the results from COPASI and Amici. Hey, we've got 3 now. Okay, by the way, I mentioned that libSPML SIM had bad results. The bad results from that were exactly the same as the bad results from VCell. So I suspect that it's a similar bug that one has, but I did not pursue that one anymore. The other one was Tellurium, and again, Lucian, who's also, I think, in the audience, helped me with this. I really got bothered because the reason was CVOD was failing because this model is not stiff. This model is very non-stiff. And by default, Tellurium runs the CVOD stiff version of the solver. And it should use for this problem, it should use the non-stiff version of the solver. So I decided to write a little Python script and call Tellurium myself rather than use run biosimulations. And in that little script, I just tell Tellurium to use the non-stiff version of CVOD, and then it works perfectly fine. It produces the same results. And then there are four pieces of software now that could reproduce all the results. And here they are side by side so that you can see that visibly there is no difference whatsoever in their results. They're all running. And by the way, they're now running with different integrators with different tolerances. So this is reproduction. This is really now using different methods that come to the same conclusions. We could have a discussion about this. I did discuss with Lucian why CVOD was not converging. I mean, the reason is that the CVOD doesn't automatically switch like LSODA does. And when models are not stiff, the stiff solvers are not very good for them. Now, Amici worked, and that's because Amici uses a default tolerance of 10 to the minus 16. And that was good enough to make the non-stiff version work. But the default values for Tellurium were not good enough. That Tellurium uses for CVOD were not sufficient. But anyway, I showed all 4 can use the same SPML file and can produce the same results. In fact, the same OMX file. This is the other variable, so they're also equal. I think I'm getting close to the end. And then the final R from FAIR is reusability. So I wanted to show how can I reuse this model now for other purposes. And so this was easy. Now that I have the model, I can just try to do something else. So I tried to find out what parameter values. So it turns out that some parameter values generate multi-stability. So the models are multi-stable. They can have 2 different stable steady states. And that's actually what happens in the pattern. One of the cells gets into the other steady state. And in fact, Ingoglia did study that a little bit in a 2004 paper. But I decided to ask, well, let's just do now, rather than do an analytical study, let's just do a numerical study and trying to find out if I see differences between what sets of parameters generate by multi-stable, generate versions that have multiple steady states versus those that don't. So what I did is I want to generate a random set of parameter values, P of those. And then for each set, I generate another number of initial conditions, in this case, a number I of initial conditions, and then find out for all of these samples, which ones produce more than one steady state. So the number of simulations is P times I. So if I'm going to do, well, we'll see in a moment. So I can encode this in COPASI very easily, and the parameter scan function. I say, well, let me do 5,000 random samples of the 48 parameters. So they are not here, but there should be 48. They don't fit in the screen. So I'm showing the first two, and then there's another 46. So essentially, this does one random value sampled for each of those parameters. And then for that combination that is sampled once, I now sample 15 times random initial conditions of all the variables. So for each random sample of parameter values, I have 15 random initial conditions. And then I look at the set of 15s and I ask, are they all the same or are some of them different? If some of them are different, I've proven that there's multi-stability. So this is a numerical approach. It doesn't prove that there is no multi-stability when I only find one value, but when I find more than one, I prove that there is multi-stability. So I'm going to have some error here, but by the way, these are 75,000 simulations right there. So I used Cloud COPASI, which is a system we created to basically, it's a web front end to send COPASI files to be run on a cluster. So we used our cluster here. And of this 75,000, 1,600 did not find a steady state at all. I suspect they were oscillating, but I don't care. I mean, those are out of the picture. 3,387 have at least one steady state. And of those, 498 did show to have more than one steady state, which is about 1 in 10 of all of the random parameter sets. So 498, it's a little bit more than 1/10 of 3,300, but it's close to that, let's say, ballpark. It's about 1 in 10 produce multi-stable systems. Now, this is a lower estimate because, of course, I may be missing. I only do 15 samples. Maybe if I had done 20 samples, I would find out a few more out of these 3,000. So this is a numerical approach. I'm just proving that I can reuse now the model to do something else that they didn't study. And then once I look at these, I can ask the question, well, what's similar between these 498 versus the other ones? What's important in them? And the only thing that I found out is that this parameter KCN-PTC has the median value of these guys is about 5 times larger than the median value of that parameter in those guys. That's really the only one that has a large difference. There were also differences in these other 2 parameters, but they were, and these other 3 parameters, but those differences were much less than 5-fold. They were like 1.2-fold and things like that. So at least one parameter seems to be interesting. So I picked I picked that parameter and another one, the next one in line, which was that, and I plotted all the parameter sets that I sampled. Those are here, and the distributions for those parameters. So the CN-PTC and the kappa CN-EN and their distributions here. So this is the one that has the largest change, 5-fold. You could see the blues are the ones that I did not prove, that I only found one steady state. And the oranges are the ones for which I found more than one steady state. So you could see the distribution for the more than one steady state is skewed to the large values for that parameter. Slightly skewed for larger values as well for that one, but it's really not that significant probably. I mean, it's hard to see that. But you can see that there's many more orange circles in this end of the parameter space than on that end. So probably large values for these parameters tend to be required for multi-stability, and those parameters actually govern feedback loops, and therefore it all makes sense. So I showed that I could reuse this model to do some other study that was not done in the original paper, just by trying to interpret that and using some of what Ingoli studied in the 2004 paper. There are, in the network, as I show here, this is a portion of the network, there are 2 overlapping negative feedback loops. They're negative in different ways, but if you think about them, PTC is that thing as well. This is the total amount, that's the PTC, this one with the other 5 summed up. So there's a negative feedback loop on them, between them and the things that they affect. And there's also the same thing this way. So there's 2, PTC has a feedback loop on itself through 2 different directions. And the parameters that were shown here that are different are part of those 2 feedback loops. So that's one interpretation. Okay, just so really to the end, this is the last slide, is lessons learned. What did I find out with all this exercise? Well, the first thing, as you can see by allowing me to provide a whole hour of a seminar, is that this is not easy. Reproducing legacy computational models is hard. That's the first take home. And I really appreciate the curators of biomodels and other repositories, because it's really hard work to do this. If you want to make your models fair, that is usable for many decades to come, don't save them in a proprietary file format like the Ingenu file format or some MATLAB code or whatever. Please save them in a format that is a standard that many applications can load, and for which is well defined, that people can find the definitions and later on write software for those definitions, even if all the software has disappeared. And also try to have that model encoded in that standard format, uploaded to a repository or a model database. So that will enhance the chances that other people can find the model. And when I mean other people, I'm talking about other people 25 years from now, or even 50 years from now. They might want to load this model. I've looked at models much more than 50 years old. And this would ensure that they can do something with them. Well, some smaller take-homes, be careful when you use CVOD stiff method, because that's unstable when you have very non-stiff models like this one, where everything's on the same time scale. The stiff methods are not really that stable. And that's why it was so important for Linda Petzold to develop LSODA, the heuristics that allows you to switch between stiff and non-stiff methods, because that is very, very robust. Immediately, any model you throw at that thing will always work, because if it's too stiff, if it's very non-stiff, it goes to the non-stiff method. That's why COPASI worked straight. Well, the other thing is there are issues with the numerical methods that make interoperability harder. And that's, for example, the case for CVOD, but that's also the case for what I mentioned underneath, which is the numerical errors that you can get with things that you never thought about, because computers work in weird ways. Runbiosimulations.org should find a way to allow tuning of numerical methods, because I tried setting different values for the tolerances, but it wasn't possible. I've already told some of the people that run biosimulations about this, but that's one issue. Hill equations, again, there's a problem. And then our last take-home message is never, ever do hexagonal grids. You don't want to do them. Believe me, if you have to write the code, you don't want to do them. I need to thank a number of people. Lauren, for bringing my attention to this whole problem of this model not existing. When she came around, I really thought that, oh, I'm going to find it in biomodels. I'm sure it's there. This is such a famous paper, but that wasn't the case. Well, Frank Bergman really is a star. I mean, every time there's something missing in BASIC or that doesn't work well, he fixes it in less time than you can think. Usually by the time I wake up next day, it's already fixed. So he's really a hero. Jon Moraro and Lucian for helping me finding out the issues with vCell and Tellurium and running the model through those two software. Aaron, we've been discussing a lot about these things, and we're actually using this model for other purposes, which one day maybe we will talk about. And also Michael Blinoff, who actually ran a special issue at Frontiers, and it was only because he ran that I actually ended up writing this as a paper. And that's it. I think I've gone over maybe a little bit over my time, but thanks for your attention, and happy to answer any questions.
Thank you, Pedro. That was fantastic. So, questions? Herbert?
Yeah. So I got to go to another meeting, but I've got a couple, if that's okay. First thing is, how long did it take to do this?
On and off, about a month and a half.
Do you think it's six person weeks?
Probably.
Wow.
Maybe more, because I mean, once I started hitting problems, like that was to build a model and get it running in COPASI. Then I started all the issues, right?
Yeah.
What's the problem with VCell? What's the problem with tellurium? Finding out those things, learning how to run them in different ways, etc.
So if you add up the cost, right, of your wages and so on, it's quite significant.
Yeah. That would be
Right?
expensive.
Yes. The other one was you mentioned SediML. Is SediML enough to reproduce this model?
Not the sampling.
No,
it was enough to reproduce a time course. That's what I encoded in SediML, was just run a time course for this specific parameter set. I don't think I didn't look carefully to encode the sampling in SediML, but I don't think so.
Yeah, you're probably right. Yeah. Okay. And the last one is there are a lot of people using Python now. There are a lot of simulators that use Python, and every Python API is different. And this is something I've brought up a number of times. I think there's a case for us to get together to try to standardize a Python API, because one day COPASI will cease to exist probably.
But
then we won't be able to run your code.
But the issue is not COPASI. This is that's a very important point that I made, and maybe I need to say it again.
Okay.
The important part here is the SBML, because
but all the sampling you did is not reproducible.
Yes, because SediML isn't up to scruff yet. There should be a standard description of that.
Oh, I see. So
because once there is a written definition of those formats, and published, then someone else much later on can actually write their code to do it.
Because it'll generate the appropriate Python for the appropriate
tool. I did that. I mean, I did not need Ingenu. Now I have it. Maybe one day I'll try to run it and see if I get exactly the same results. But by the way, I made a purpose of not trying to get Ingenu, because I really departed at that point to, can I really do this?
I mean, Frank and I have been talking about a standard API, and we produced one way back like 15 years ago, and we're thinking of resurrecting it. But anyway, I see your point, though. Okay, yeah, thanks. That's it. Thanks very much, Pedro.
Sure.
Other questions?
By the way, let me just make a statement that I'm not sure I did. I put that in the paper because I think it's only fair. There are a lot of critiques about this paper. Reproducibility is not one of them. They actually, I mean, as you saw, there were things missing that I had to guess, but they were minimal compared with this is really a complicated model. They did a great job, and I'm really not a fan of Nature and commercial publishers, but I have to say they also did a good job of still keeping the supplementary data, because I tell you, papers that are 24 years old in many journals, they change the website. Once they change the website, all the supplementary data gets lost, because it's only the PDF. It's the only version of record. The supplementary data is not version of record. The journals don't treat it as, they treat it as a little bit of dessert they're giving us. So I have to say this. I don't like their conclusions. I don't think this shows any robustness whatsoever. But I think they did a great job with this. Their software was actually looked good. I never ran it in those days, but
I've got a bigger picture question. You said it's a classic model, right? So are there a lot of other classic models that are yet to be added to the biomodels repository that should be?
Oh yeah, I just encoded a couple of them. Nothing like this. Much easier things to do. Things that you do in a couple of hours. Yeah, there's many. But I would say that any of the paper that describes a model that has more than 2,000 citations, you can call it a classic.
Should there be like a concerted effort to get those papers onto biomodels, kind of use them as kind of a showcase to get people to actually upload theirs? I don't know.
Yeah, I think that would be nice. I mean, I've heard many times the idea of getting students to encode these things and do it as summer projects and stuff like that, and this happens. So anyway, Jim Fader seems to have a question there.
Yeah, well, I have multiple questions, but I don't. So I've known you since about 2000. I don't remember exactly the first time, but I don't feel like you have aged. I don't understand how this is possible.
Well, I
But
could probably say the same thing.
yeah, I mean, yeah, if you look closely, you could. We keep active, I guess.
Yeah.
So the question I have is did you try to do this with a square lattice after the hexagonal? Does it change the findings drastically?
No, it doesn't. It doesn't change the findings much. There were a couple of papers that did that. I think Eduardo Sontag, two of those are from Eduardo Sontag's group. They used a square lattice. It doesn't have a big effect. By the way, in the second paper, they try to justify why they use hexagonal. They show images of these cells, and they're kind of, with a little bit of imagination, you can think that they're hexagonal, but they are actually much more irregular than that. But hexagonal is a really bad idea.
Yeah.
Hexagons, well, they pack well.
Yeah, they pack well, but you know, you pack them in 2D, but they actually need to be indexed by 3D. They need 3 coordinates to be indexed because they have 6 sides. Well, they have, yeah, so they are oriented in three parallel directions. So the people who do games have worked on that as well. And when I looked for code, I thought that there would be good algorithms to do this, and it's not. It's like I found out blog posts and discussions on threads that were like, really, this isn't a work of the devil, making hexagonal arrays.
Other questions?
Okay.
Yeah, so I guess we can thank our speaker. Thank you, Dr. Mendes. That was a great
Thank you very much for
presentation.
having me.
Great work. And with that, we're 11 minutes over time, so I guess we can call it unless somebody has any last minute questions.
A real quick one. Do you have a listing of future? I know future talks. I know the next one that was mentioned was a PyPesto demonstration or
Yes.
discussion. Or is it? I just didn't see a schedule somewhere. I was going to
July-
share it with.
July 12th?
Okay. Is there a
Yeah.
website?
Yes, there is.
Okay.
Can we hold on a second? I will put it in the chat.
Oh, you're beating me to it. I couldn't shrink my screens quickly enough to get there.
There you go. It's in the chat.
Thank you.
Beautiful. Thank you so much. I'll
All right.
try to get the word out.
Cool.
Thank you. We
Thanks
appreciate it.
again. And that's it. So everyone have a good day.
Thank you, everyone.
Thanks.